Good evening again, and welcome to this webinar series. I assure you that it's going to be a very exciting evening, mainly because I'm a biologist who gets excited about the smallest of scientific discoveries. And having been in Genotypic for the past 3.5 years has kind of added more layers to it, mainly because of the technology that I'm currently working with and the company is involved in, which is the Oxford Nanopore Technology, which helps us sequence genomic data and also analyze it, apart from the other platforms that Genotypic has already been working on. So as fellow scientists, I mean, this congregation is included, and it has several people from different diverse backgrounds, be it biology or maybe some from corporate backgrounds. But I really hope that this webinar is going to be helpful to each one of you, and it will help you answer several scientific questions, which probably are at the moment difficult if you are still working with technologies that don't cater to very customized needs. So let's begin. So the title of today's webinar is From Raw Signals to Epigenetic Insights. We as biologists do understand that there are several layers of science to any biology, and some of them are quite obvious. And if you consider genomics, sequencing ATGCs is kind of routine. But to understand what is actually also on the side or provided as a side dish is very important, because it plays a definitive and very significant role in how biology works and also organisms function. So today's webinar will help us uncover Methylomics and provide epigenetic insights using a very advanced and sophisticated technology, which is the Oxford Nanopore Sequencing. So what is epigenome? We all know that the genetic material most organisms have, and in some cases, and there are a few
exceptions to hide the.
Yeah.
Thank you so much.
Yeah. So what is epigenome? Most of us know that the genetic material is DNA in most cases, except for some organisms where RNA is the genetic material, like in viruses. So this DNA gets transferred, DNA or RNA gets transferred to the next generation. But what determines the proper regulation or expression of this, of every gene, is what is called as epigenetic modification. And this can be of different kinds, wherein your DNA could be methylated, it could be phosphorylated or acylated. And in this case, there could be different conformations of the DNA as well, where it gets folded into complex scaffolds. And like we know, it does get folded into chromosomes. And this folding depends on the kind of modifications that are there on the histone proteins. So if you consider a single cell, there might be several writers, erasers, and readers that will kind of check this specific mechanism to ensure that at a given set of time, there are some genes which are expressed and some which are shut off. Like in this case, if I'm talking to you, it is ensuring that my endocrine system is working efficiently, just so that I'm able to provide you or the right information and also help you understand correctly. So it's a kind of on and off switch, and depending on the kind of modifications, your genes get expressed. It's very important to understand how the epigenome functions. Now, people have often wondered whether we inherit epigenome, and it is true. So we all know that the genome as such is inherited from either from both the maternal and the paternal side in humans. And there's a part of the genome that is having these modifications. So it is a known fact that we do inherit the epigenome. And there's kind of imprinting, which is like very significant methylation marks that are present on the genome, which get transferred down the line into the progeny. But in addition to this, there are several other environmental factors that may also play a role in the way these epigenetic regulations come into picture. In case of maternal nutrition, the food that an expectant mom eats might also help, or it might actually lead to some changes in the way the genome of the baby is affected. And apart from this, we see that there are several environmental factors, the lifestyle that we have, or the very fact that there's so much pollution now in this world, that all these factors are in some way contributing to the kind of epigenome we have. And it is constantly changing the methylation marks or the or the other epigenetic marks that might be regulatory in nature. We all know that in biological aspects, when an embryo is formed, there is certain part of the genome that gets demethylated. So this complete genome undergoes kind of a reset, but at different stages of the embryo formation, there is certain percentage of methylation that still remains. And this is what gets transferred or inherited by the progeny. All these other environmental factors that I mentioned do affect us in different ways, where there are some lifestyle diseases which have come into picture. It does set in early aging because it's been known that more the methylation, more the age of an individual is, it be it a human or any other organism. So epigenetics is important to understand different aspects of biology and how the environment also plays a role in bringing about this effect. As I go further into the talk, I'll just hit upon a few of the clinical outcomes later and how this advanced technology that I'm going to talk about will help us deduce this information. Now that we have talked about epigenome, there's also a concept called epitranscriptome. Transcriptome is what is expressed. It forms forms all the mRNAs that are present in the organism and are leading to protein formation, and thus the organism is functioning. But this mRNA also undergoes epigenetic changes, or it does undergo modifications. And till date, several types of modifications have been identified. Here, I've just highlighted a few. In the guanosine, there is methylation at the 7th position, and also M6A, which is more predominantly observed. There's also M5C. And like in the genome, even in mRNA modifications, there are certain writers, readers, and also erasers. So depending on the state that the cell is, different players come into picture in this case. It also determines whether your mRNA is going to be stable, or it will be recycled, or it will be sent for a mechanism wherein it gets degraded. So that way, these modifications actually govern how your expression status is. So does the epigenome and epitranscriptome affect us? As I have said in the previous slides, yes, it does. And it brings about a lot of changes, specifically now in this environment. We see a lot of it being contributed by the epigenome and the epitranscriptome. And if you want me to put it dramatically, genes just load the gun. It's the methylation that pulls the triggers. So I've listed down a few of the effects that can be part of this. And it's mental health, where it's been observed that the kind of stress that we undergo brings about methylation changes in our genome, which affects health, mental health. Cancer, it's a very well-studied field, and there have been several studies which have shown that methylation status does determine your cell cycle and thus could lead to cancer's tissue formation. Aging is one criteria that was already discussed before. Reproductive health is affected by methylation. Crop improvement for all those who are into agricultural sciences or working with plant model systems. This is important to understand because there are several environmental factors which are affecting the current plant health, and understanding these changes will actually help us improve our crops. Heart health, like I said, many of the clinical aspects can be contributed to the epigenomic changes, and there's diverse biology that gets affected by the methylation status of a specific organism. In order to understand the methylated state of DNA and RNA, several uh, technologies have been attempted and have been in practice. The more traditional ones have been the whole genome bisulfite sequencing, which has been prevalent for a very, very long time. And then there's reduced representation bisulfite sequencing. There are also other mechanisms, other technologies which are existing in order to identify these specific changes in any organism, in the genome of any organism. But they do have certain setbacks, or they're not very uh, it, they're not very whole. As in, there are certain issues which come into highlight when we talk about these technologies. If you talk about bisulfite sequencing, the uh, the processing involved is so excessive that it does lead to a lot of DNA being degraded or being of bad quality. And the percentage of the methylated genome that gets represented in such cases is a very small percentage. So all in all, we are unable to capture the entire methylome of any organism using these technologies. But what if I tell you there is one technology which, without any processing of your nucleic acid, can actually provide you a more holistic picture of what the methylated status is. And in addition to that, it also provides you additional information. So there's just one single reaction that can help you deduce many aspects of the genome and give you several information. And that is one technology which I've just tried to compare here with the short read technology, and it's the nanopore technology. So if you consider the short read platform, nucleic acid preparation is extensive. The library preparations involve PCRs, and that could lead to some kind of a bias. And the sequencing is often extensive. You need a lot of depth to reach a state where analysis of this data will provide you useful information. But in the technology that I am talking about today, we don't need any of this extensive nucleic acid preparation. It's a direct library preparation. It's a single molecule sequencing platform. And the sequencing is also not long drawn. We can generate data quickly. And there are standardized analysis pipelines, which will help us understand the information. So this is the nanopore technology that I am talking about. And this slide in particular explains how the technology works. I think there have been several webinars conducted before, and many of you might be aware, but this is just for the information of students and other people who are not aware of this technology yet. In this specific platform, there's a matrix on the flow cell, which is embedded with several nano-sized pores. And the protein complex is made in such a way that once the library is made, it has an adapter sequence which goes and tags along with the tags that are provided on the matrix, and it helps unwind the DNA. So your double-stranded DNA from the library passes through the pore because there is a difference in the ionic current, and that is the reason it just goes through the pore. And once it goes through the pore, there's a small reader which kind of detects the electronic signal that comes along with each of the base when it passes through this pore. And the signal is then converted to the base that has been detected. And for this, several algorithms were developed, and it's a very standard algorithm which kind of produces the final sequence of the genome or the transcriptome that is going through this nanopore. Uh, the There are several benefits of this specific technology because the workflows that have been developed to make the library are very simple, and the kind of lengths that we get are also really incredible. We have been able to get DNA lengths of about 1 MB as well. So that way, assembly procedures during analysis are much easier because the longer the fragment of DNA that you're able to sequence, better the assembly or easier the assembly is. So how does any modification that might be present on your DNA get detected? It's because the normal ATGCs have a very specific signal signature when detected by the reader. But if the base is methylated, there's a deflection or there's a change in the signal. And this is captured by the signal detection mechanism, and it gets read as a modified base. There are several algorithms which further then act, or tools which can then deduce the kind of modification that is present on the nucleic acid. So why would one choose nanopore technology for such an analysis or such a study? Mainly because it's hassle-free. You don't have to worry about any nucleic acid preparation run through huge protocols, and which might actually not give you complete information. This technology actually helps you provide comprehensive data, wherein a single sequencing reaction will provide you information on different aspects. And this is native molecule library. It provides a more even coverage for the genome, and there's no GC bias because there are no PCRs or any conversions involved in this technology. We have compared the data for short-read platforms with the data generated on Nanopore platform, and we find that the CPG positions that were called were at lower depth, so we did not need extensive depth of sequencing in order to get this information. So haplotype tracing is also possible because you can have some standardized pipelines which will help you deduce this. Then, because it's a native molecule reaction or sequencing, it does provide a lot of reproducibility. So that way, since there are no conversions involved, there's no excessive processing, there are no batch effects as well. And like I said before, this data generation and analysis is considerably faster compared to the short read platforms. So this slide actually tells you in detail the other types of informations that can be deduced using one single sequencing data which gets generated from any sample. So if one has generated data for, say, any plant sample, then using the data, we can get the methylated status of the genome. We can do variant calling. So single nucleotide polymorphism detection is possible from the same set of data. We can also analyze the structural variants. And this is possible only because Nanopore provides long reads. And these long reads help us kind of assemble data more correctly, because sometimes with short read platforms, the repetitive regions may not be resolved properly. But since we are getting very long reads, the repetitive regions, the methylated status for these regions can also be identified. Phasing is also possible with this technology. And like I said before, assemblies are much better because we are able to generate long reads. So the gaps that come in, or the resolution with respect to the repetitive parts of the genome, are better resolved. We have had the fortune to collaborate with a fellow scientist who was involved in a study that was dealing with diabetes and how methylation status affects individuals before at a certain stage of their age, and how these then turn into the diseased conditions. So we worked very closely with him and then were able to provide really good information, scientifically correct information as to what might be the signatures that could determine a pre-diabetic patient from a normal or a diabetic patient. So 6 years down the lane, if a person has been identified with certain methylated signatures, those can be used to predict whether he or she will be diabetic after a certain time. And so these visuals that you see are from that data. And I'm also happy to say that this data could be validated from the metabolomics data that he had generated for his other part of the study. So all in all, since this was the first Asian or the Indian genome that we were working with in our firm, we were also able to validate it using another Asian genome. And we found that the nanopore data had about 95% overlap with the public domain short read platform. So there are no errors that we could really identify, and all the methylations were called correctly. So there are certain perks if you talk commercially on the Nanopore platform, mainly because if you have short read data, you would need extensive depth, say about 30x at least for a human genome in order to identify methylations. But in Nanopore, you don't need so much depth because of the longer reads that we generate. Even about 15 to 20x data can help you identify these modifications and provide correct information. And like I have already said before, repetitive DNA is an issue with short read platforms, but the long read platform does take care of this. Structural variant detection is very ambiguous when you consider short read platform, but it is quite precise considering the long reads that get generated using the Oxford Nanopore technology. And the other parameters that are mentioned here, I've already discussed, so I think I'll just move forward. Some of you actually might be working on some of you might be working on clinical aspects of human biology and might have to deal with certain samples which are really difficult to work with. And cell-free DNA has been known to be a prognostic marker in this case. It is also important to understand different cell types depending on the different markers that can be identified based on either transcriptome expression or the methylation status. And people worldwide have developed atlases which provide the specific or precise information on different cell types that have very signature marks for methylated genes and also genes that are expressed. So there is nanopore technology actually can help you understand this in more detail because there are pipelines, and since some samples may be very limiting, like the cell-free DNA, which is really difficult to work with, it's just the freely available DNA in any sample, be it blood or any clinical sample. So depending on the kind of modifications that get identified in this type of sample, you can deconvolute the data using a specific tool that has been developed. And we have standardized this pipeline in our firm, which is called the Nanomics tool. And it helps you understand the types of cells from which the DNA is arising. So depending on the methylated status or the cell cycle state, you can then correlate the data and understand whether there's any cell type which is originating from a cancerous tissue. So this figure here just is kind of representation that provides information on each of the cell types based on the methylation status of the genome data generated from a cell-free DNA sample. Uh, I did talk about the human epigenome in great deal, but if you consider biology, all of us as scientists are working on some model system or the other. And epigenetics has played an important role in kind of changing how an organism behaves, or also to a large extent, it has played evolutionary significance. This slide in specific tells you that how transposable elements, which kind of have the capacity to jump from one part of a genome to the other, are able to uh, direct evolution. If you look at the distribution here, this is just a percentage of the genome that comprises the presence of transposable elements or contributed by a transposable element. In many systems, specifically fungi, these transposable elements are not allowed to replicate much because they have certain genome defense mechanisms which help prevent activation of these elements. But there are others like if you consider humans, the lungfish, maize, all of these organisms have a huge representation of the genome which which come from transposable elements. And if you talk about fungi, they have these genome defense mechanisms which lead to transition mutations and also cause methylation of the transition mutations that have happened, thus silencing the transposable elements. So all in all, there are several biological interactions that are kind of directing the course of biology and evolution. There are several other phenomena like host pathogen interaction, which get affected by the epigenetic mechanisms that are present between the co-players. So apart from human, we need to understand plant methylome as well, mainly because they are our lifelines. They are the ones that kind of provide us oxygen. So I think it's important to keep in mind that understanding plant methylome would be an important aspect. So plants do have methylated genomes, and if you consider certain grain types like wheat, which is a highly polyploid grain, and we do understand that there's duplication of the genome several folds. But does this duplicated genome get represented in its transcriptome as well? Is it multifold increase in the genes that are all present in several copies of this genome? Or is there a mechanism that is making sure that only certain sets of the duplicated genome or the multi-copy genome get expressed? And this comes into highlight if you consider the methylated status of the genome. And we have, in partnership with collaborators, tried to understand the different significant contexts in which a plant genome gets methylated. It's also important to understand that plants have a symbiotic relationship with several organisms which contribute towards its health, be it the rhizosphere that it's present in, or the kind of pathogens or the commensals that it harbors. It also depends on the interaction that it has with the pollinators, which are very important because if the pollinators don't get attracted to a plant, then there will definitely not be any fruit formation. Therefore, these interactions are guided by the genome, which could have certain signatures that at a certain point of time might lead to certain specific changes in its volatiles or the way it interacts with the other living organisms in the rhizosphere or the phytosphere. So animal methylation we understand, but in plants, it does differ to a certain extent. So definitely plants and animals, even though they are biological organisms, do differ in the context of its methylation. In plants, we have found that there is predominantly CG and CHG context of methylation. Even CHH methylation is found predominantly, whereas in animals, we do find only the CG type where if you look at the enzymes involved or the mechanisms that are present in these two different biological setups, they are slightly different. In plants, it has been identified there are small RNAs which do guide methylation, and this is in case of animals absent. So there are very specific mechanisms that come into picture when you consider plant genomes. And we did have, again, the good fortune of collaborating on a very specific plant project, wherein we were able to, after a lot of experimenting and detailed analysis, able to identify precise methylated status of different sample types that were generated in the experiment, and using the standardized pipelines that were designed by a very talented team of bioinformatics in our firm, we were able to get the detailed picture of the CHH, the CHG, and the CG type of methylation for a plant genome, and this will soon be published. It was a lot of interaction, and through this collaboration, we were able to learn a lot of biology, as well as how different bioinformatic analysis can kind of change the picture or help us understand the picture better in biological organisms. Apart from plant, methylation is also present in bacterial DNA. It's slightly different, although it has been thought that for a long time, it has been thought that bacteria do not have methylation mechanisms, but more recent studies have shown that these are in fact present because it provides them a kind of genome defense mechanism against phages, which would then kind of create a havoc if they were to enter the bacteria and play with the life cycle of this specific bacteria. The restriction modification systems that are present in bacteria have also been utilized a lot, and technologies have been developed using this specific mechanism to come up with molecular biology kits that can help us understand specific questions. Apart from this, some of these bacteria might be human pathogens, animal pathogens, or plant pathogens. And the role played by methylation in this case is very significant because they have the capacity to alter their own as well as the host genome and target methylation, which will help them kind of interact better with the host to promote their own well-being and cause the pathogenesis. Uh, I think some of you and many of you might not have heard about some mechanisms that are present in fungi, and all of us work with different model systems, but they are very interesting, and fungi play a very important role in our lives, mainly because they are the ones who interact with plants. They help the plant health. They have been able to ferment food for us, provide us wine. They are the ones who gave us our antibiotics, and they are food producers and also biological control agents against many insect pests and also bacteria. But they also call allergies, are very bad pathogens, and also affect us in other possible ways. So in order to understand these very interesting systems, we need to go into detail about how the genome is made up. Some of them may have transposable elements, while some may not have. Some may have gene silencing mechanisms, while the others may not. But if they do have any methylation mechanisms or how these modifications are made, it is important to understand them so that we are able to better control these organisms, maybe come up with better food fermenters, come up with fungi that will provide us better nutrition when used as food products, and also help us understand how pathogens interact with the host environment so that they cause more disease conditions. For some of you, it may not be possible to kind of, if you consider financial aspects, or also for want of interest, you may not want to understand the whole genome perspective of the model system that you're working with, or you may have very targeted regions or genes of interest that you might want to work with. And if you consider the other technologies, anything if you want to have a targeted approach to, there will be some kind of a processing involved, PCRs and other such things. But if you use PCR based methods, then all these methylation marks or modifications will be lost and the information will not be obtained. But there is one specific technology where, without any physical intervention, just using a bioinformatics approach, you can actually get all this information and you can make it targeted. It's a PCR-free targeted sequencing mechanism. And what I'm talking about right now is the adaptive sampling mode of nanopore sequencing technology. The way it works is when a nucleic acid passes through the pore, the the system or the base calling system reads the first 400 bases of the sequence. If you're able to provide a reference, it just tries to match the 400 bases to the reference. If it matches, it lets the molecule pass through further and sequence it. But if the sequence does not match the reference that you have provided, then it just reverses the current and rejects the strand or stops sequencing. That way, it provides an enriched data for the gene of interest or the region of interest that you really want to study. There are certain uh, there's certain information that you need to understand about this, because it's a random mechanism, because you cannot direct only the targeted regions to go through the pore, right? So sometimes you might end up generating data, but your gene of interest targeted data would be little, but that may not always be the case. So there will be certain tweaks that, when applied, would help you generate more such enriched data, and analysis of that is easy, and you would be able to get targeted information that your study demands. All this while, I did talk about DNA sequencing, but there is one revolutionary technology that is very specific to Oxford Nanopore, and that I will talk about in the next few slides. It's a known thing that RNA sequencing is not very easy. Uh, to think about it, RNA itself is really difficult to handle, because the entire environment is laden with all kinds of RNases. So handling the nucleic acid itself is kind of a little tricky. But once you're over that problem, if you want to sequence and understand the epigenetic modifications that the specific RNA has or mRNA has, then it would be really cumbersome to do that. It's known that there are several modifications identified in RNA, and these do help direct the biology, wherein any modification that is on the mRNA will either target it for the degradation mechanism, or it would let it get expressed. But there are certain modifications which, when occur on certain parts of the genome, will then lead to silencing of the genome in certain regions, which may not be the ideal case for any biological system. So in order to understand the specifics or the molecular aspects of RNA methylation, it is important to come up with a method that does not include excessive processing, because all the methods that are prevalent today, or which have been worked upon, are very targeted. So if you try and understand one type of modification, then the other modification information is lost during the processing that is done in order to come up with information for the specific RNA modification. It is also not comprehensive, and whatever technologies have existed in the past have provided minimal resolution. So the revolutionary technology that I hinted upon was the direct RNA sequencing using the Oxford Nanopore platform. It's unique mainly because, in this case, this is the only technology where you can directly sequence RNA. You don't have to go through the hassle of using reverse transcriptase to come up with cDNA, and also the depth that you need for sequencing is not excessive. short read platforms, even if you want to do a transcriptome, will always need a lot of depth to bring out the information of the transcriptome. But in this case, with little data, you will be able to get all this information. So the way it works is that you extract RNA, you get the poly-enriched RNA, and then you ligate a poly-DT adapter, which just puts a tail of T's at the end, which binds with the poly tail. And then you ligate the adapter and load it on the RNA flow cell. And once the sequencing is done, depending on the algorithm that is used, it reads all the bases. So you get the sequence information along with any differences that the sequence has generated in the signal. So using this platform, you're able to generate raw reads, which if needed can also be rebased call at higher accuracy in order to deduce more detailed or more specific information to your nucleotide sequence. The perks about this sequencing technology, I will hint upon a little later, but in our lab, we have also developed standard pipelines which help you understand the modifications on the mRNA. In this case, Eligos 2 and the mean workflows have been standardized to identify M6A methylation in mRNA and also other contexts that it might have methylation, which might not be more prevalent, mainly because it's the biology that works that way. So MEME is another tool which actually helps you deduce the motif which is targeting this kind of a RNA modification. And it's important to understand that because studies have been able to classify the motifs to the path the RNA would take later based on the modification that it has on its sequence. There are some modifications which are being worked with by the Nanopore platform, and very soon we should be able to identify these modifications as well. And here in this case, one of the ones that I talk about is 8-oxo-G, which is involved in stress granules. So once data is generated using direct RNA sequencing technology, you don't have to stop there because you don't have to go through another sequencing platform to generate transcriptome data, because all data generated using the DRS method will provide you transcriptome data. It will help you analyze the poly-A length also. Poly-A length analysis, like many of you might know, is very important because it plays a major role during development. Poly-A is something that actually guides, it decides whether the RNA will get degraded or not. And then transcriptome analysis is easy because the depth of data required for analysis is not much. So you get all the information apart from being able to identify the modifications and also the poly-A length. It's also important to understand what kind of isoforms are present or if there are any fusion genes. No other technology will help you understand these aspects, but Nanopore DRS will provide you all this information. So you're getting 3 freebies in the cost of just one sequencing reaction, which I think is a win-win situation for all of us. So I think I have touched upon a lot of technology and also hopefully something to interest all the biologists and scientists that have assembled virtually to listen to this talk. And I would like to say that there are certain collaborations which have really helped us understand biology and get a different perspective. And the technologies that we have worked with and have constantly uh, interacted with scientists or collaborators which are working with different systems, and they have helped us improvise to a large extent. So I think it's all you who have made us what we are today. And if you do have any interesting questions in epigenetics or any other aspect, please talk to us. I think we have bespoke solutions that will help you understand these questions or answer them. And we would be really excited and happy to work with you all. Thank you.